Before a vaccine is ever recommended for the public, it goes through a rigorous process to make sure it's both safe and effective. Yet many people still wonder, how do we know that vaccines are safe? In this video, we'll walk through the key steps of vaccine development, including clinical trials, vaccine licensure, and product recommendations, and we'll explain how safety is monitored each step of the way. But first, it's important to acknowledge that there are very few choices in life that are completely risk-free or offer absolute protection. Take buckling your seatbelt, for example. More than half of the people who die in car crashes weren't wearing one. While seatbelts can sometimes cause minor injuries, they remain the most effective way to prevent serious harm or death in a crash. That's why they're required in most places. Vaccines work in a similar way. They're not perfect, and they don't offer 100% protection. Some vaccines help prevent infection altogether, while others are especially effective at preventing severe illness, hospitalization, or death. Like seatbelts, vaccines carry a very small risk of side effects, but serious reactions are extremely rare. And overall, the benefits of vaccination, protecting individuals and communities from serious disease, far outweigh the risks. Whether it's deciding to buckle up or get vaccinated, we're always weighing risks and benefits. No choice is completely risk-free, but some give us the best protection possible. That brings us to the topic of vaccine safety. How do we know vaccines are safe? What standards are in place to make sure the benefits truly outweigh the risks? Because vaccines are given to millions of healthy people, including infants and children, they're held to some of the highest safety standards of any medical product. Unlike medicines used to treat people who are already sick, vaccines are preventive. That means the public's tolerance for risk is much lower, and safety monitoring must be especially rigorous. This includes quickly identifying and investigating any potential side effects and sharing vaccine safety data in a transparent way to maintain public confidence. The United States has a strong, long-standing vaccine safety system in place. Today, we have the safest, most effective vaccine supply in our history. Millions of children and adults safely receive vaccines each year, with the most common side effects being mild, such as temporary soreness or swelling at the injection site. So what does it take for a vaccine to go from development to public use? Let's start by walking through the vaccine development process. Vaccine development is a lengthy and complex process that can take 10 to 15 years or more. Vaccine development happens in stages, starting in the laboratory. It's then tested in cells or animals, and eventually humans. Vaccine licensure and determining who should receive the vaccine are the last steps. The first stage of vaccine development is the exploratory phase, which involves basic laboratory research and can last 2 to 4 years or more. During this phase, scientists are looking for natural or synthetic antigens that might trigger an immune response and help prevent or treat a disease. The next stage is the preclinical phase, where scientists use tissue or cell culture systems or animal models like mice or monkeys to determine the safety of the vaccine and its ability to produce an immune response. This stage also gives researchers an idea of the cellular responses they might expect when the vaccine is given to humans. Many vaccine candidates do not make it beyond this phase because they fail to produce a desired immune response or prove to be harmful. If preclinical testing goes well, the vaccine manufacturer submits an investigational new drug application, also known as an IND application, to the U.S. Food and Drug Administration, or FDA. Once approved, the vaccine moves into human clinical trials, which occur in 3 phases: 1, 2, and 3. Before we look at the specific phases of clinical trials, let's talk about why they're important. Clinical trials are the first opportunity to assess vaccine safety in people. Even though vaccines are usually tested in animals before humans, humans and animals are very different, and human trials are necessary to assure the safety and efficacy of a vaccine. Clinical trials depend on people to volunteer to participate. While early-stage clinical trials are small with only a few dozen participants, later trials are large and may require the recruitment of tens of thousands of volunteers. The safety of clinical trial participants is a top priority during vaccine development. Many steps are taken to ensure that safety is considered in every step, and the highest scientific and ethical standards are followed. For example, clinical trial sizes are determined by the vaccine manufacturer and the FDA, and trial size is important. Clinical trials must be large enough to determine whether a vaccine is generally safe and what side effects are common after vaccination. Importantly, clinical trial sizes are often not large enough to detect rare side effects that may occur at a rate of 1 in 100,000 doses administered or 1 in a million doses administered. To detect these rare side effects, we have many systems in place to monitor vaccine safety post-licensure. Another important aspect of clinical trials is the use of a control group. A control group is a group in the study that does not receive the vaccine being studied in the trial, but instead receives a placebo or the current standard. The control group is used as a baseline to measure the effectiveness of the vaccine being studied. Without a control group, you cannot determine the effectiveness of the vaccine, or if an adverse event occurs more often following vaccination than what would be expected in the general population. Simply put, without a control group, causal relationships between the variables being studied cannot be established. By using control groups, we're able to validly state whether a vaccine likely caused an adverse event, or if a vaccine is effective at preventing a particular outcome. The use of a control group and the size of the trial are fundamental to determine vaccine effectiveness and also to detect if adverse events which occur following vaccination are truly related to the vaccine or are coincidental. Now that we've reviewed why clinical trials are important and some of the key components that are vital to assessing a vaccine candidate's safety, let's look at the 3 unique phases of clinical trials in greater detail. Phase 1 enrolls less than 100 people. During this phase, researchers gather information on safety and the immune response. Only if a vaccine candidate is shown to be safe in Phase 1 trials will it move to larger Phase 2 trials. Phase 2 enrolls hundreds of people who generally have similar characteristics, such as age and physical health, to the intended vaccine recipients. In this phase, we are learning more about the vaccine's safety and the immune response it produces. We're also learning more about immunization schedules, like the spacing of doses, and how much vaccine is needed to elicit an adequate immune response. It's important to note that Phase 2 trials do not assess how well a vaccine actually works. Only in Phase 3 trials is vaccine efficacy assessed. Phase 3 usually includes thousands, if not tens of thousands, of individuals. Participants are chosen at random to receive the vaccine or a placebo, and participants and most of the study investigators do not know who has received the vaccine and who has received the placebo. Participants are then followed to see how many in each group get the disease, and this phase is where we get our first sense of vaccine efficacy. Assessing short and long-term safety is also a major goal of Phase 3 trials. Because of the large sample size, this is the phase where we hope to identify some of those rarer adverse events and confirm the vaccine is generally safe when given to thousands of people. Clinical trial participants are followed for a specified period of time to identify any potential vaccine side effects. Typically, this follow-up period is at least 6 months. Some follow-up periods may be longer. After that, the vaccine's safety continues to be watched by the United States' robust vaccine safety monitoring systems. But it's important to note that it's highly unlikely and incredibly rare to see a vaccine side effect occur more than 2 months after vaccination. Once these phases are complete and the vaccine is shown to be safe and effective, the manufacturer submits a biologics license application, also known as a BLA, to the FDA. The FDA's review of the BLA is rigorous, and it can take months, if not more than a year, for the FDA to approve a new vaccine. The FDA seeks the inputs of its Vaccines and Related Biological Products Advisory Committee, sometimes called VRBPAC, when considering a new vaccine. This committee is composed of outside, independent, technical experts from various scientific and public health disciplines. This panel provides input on scientific data and its public health significance in a public forum. If the FDA approves a vaccine, it then moves to the U.S. Centers for Disease Control and Prevention to make recommendations on who should receive the vaccine. The CDC seeks the input of the Advisory Committee on Immunization Practices, also called ACIP, another group of outside, independent medical and public health experts. ACIP carefully reviews all safety and effectiveness data and makes recommendations for the use of vaccines in the U.S. This includes creating immunization schedules regarding the appropriate timing of vaccination, dosage, and precautions and contraindications to vaccination. The CDC recommendation may include guidance for the use of vaccines in special populations or in special situations. The ACIP modifies recommendations, if needed, based on post-licensure safety monitoring or new vaccine data. Only once a vaccine is licensed by the FDA and recommended by the CDC is it made available for the public. So what's the difference between what the FDA and the CDC really do? While the FDA and VRBPAC are responsible for determining that a vaccine is safe and effective, the CDC and ACIP determine who should receive a vaccine and how it fits into the recommended vaccine schedule. And there may be differences between what the two groups indicate or recommend. Let's take a look at an example. The FDA has licensed Menquadfie for active immunization for the prevention of invasive meningococcal disease caused by Neisseria meningitidis serogroups A, C, W, and Y. The vaccine is approved for use in individuals 2 years of age and older. The CDC recommendation states that all 11 and 12-year-old adolescents should receive a MenACWY vaccine, and then receive a booster dose at age 16 years. The CDC also has meningococcal vaccine recommendations for those at increased risk of disease. As you can see here, the FDA has licensed the vaccine for those 2 years and older, but CDC only recommends the vaccine for those in specific age groups or those at increased risk. In this video, we've explored the key steps of vaccine development and how clinical trials ensure they're both safe and effective. We hope you feel more confident in vaccine safety after learning about the rigorous processes vaccines must undergo before they're made available to the public.